Welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Allison Waters will present Optimizing Deep Brain Stimulation for Obsessive-Compulsive Disorder. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of over 190 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has had such great impact is because we do not limit our focus to one illness or condition. Since 1987, the Foundation has awarded $450 million to fund more than 6,500 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Allison Waters. Dr. Waters is a clinical neuroscientist at the Icahn School of Medicine at Mount Sinai, New York. She was a 2019 Young Investigator. Our webinar will begin with Dr. Waters' presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many questions as possible in the time allotted. And now I'm pleased to introduce Dr. Waters. Allison, the floor is yours.
Thank you so much, Dr. Borenstein. Jeff, it is really a pleasure and an honor to be with you, and also among the many other speakers in this series that I've enjoyed so much. I don't have any financial conflicts of interest to report today, but I do want to acknowledge the funding bodies that have supported this work, including the BBRF. This slide's usually saved for last when academics present, but we've started to adopt the practice of acknowledging our colleagues first. Why is that? Because we're working on brain implants, really on the frontier of brain-computer interfacing to treat psychiatric illness. This is true team science. If you've ever wondered how those kinds of innovations come about, this is what it looks like. It's a big team. All sorts of different disciplines represented here. Clinicians like psychiatrists, psychologists, neurologists, neurosurgeons, nurses, but also neuroscientists, engineers. And we're at an institute that needs to support a very significant infrastructure to help us talk across all those disciplines. It's a really interesting challenge. And we make for a very large dinner reservation here in Hell's Kitchen, New York. And that's saying something for Hell's Kitchen. So one piece that's missing perhaps from this image is the patients, who are in many ways our collaborators. I mean, just given the nature of the work that we do, they become an essential source of insight. And I happen to know there are several patients in the audience today who've received brain implants and worked with us in our research study. So I thank you for reaching out, and I am tugging on my earlobe at you right now, just as we said. I've been thinking a lot lately about a piano tuner. I think I really like this metaphor. for what we do, because the metaphor puts the patient as the musician and sort of centers their creativity and their action, whereas we're on the side there to address more like the dissonance in the system, to tune it up, but not to play the music. It's still your song to play. The brain is not like a piano. The brain is unthinkably complex. So where we derive hope is really in how these new methods emerge to make our neurotechnologies better and better. I'm going to focus on the new emerging methods in my talk today. The things we're doing at the center in an effort to enhance and disseminate this life-saving treatment. So over the course of my talk, you're going to see how we came to think of OCD as a candidate circuitopathology, with the kinds of symptoms that can be addressed with precision electrical neuromodulation of brain circuits. And it's my hope that you leave this hour with new ideas, new knowledge about why we implant the device, where we do, how we do it. And then in the last quarter of the hour, I'm going to share with you some of my excitement about a sort of new frontier, new technology that we're developing, electrical mapping of brain waves, where the optimal target tuning for the device is coming from the brain's rhythms. So that's where that tuning metaphor really comes to life for me. I'm going to begin this story today by introducing my colleague, psychiatrist Martin Figuet, and one of our patients, Matthew. Matthew is the subject of a documentary. So here he is describing some of the symptoms of OCD that he wrestled with.
I have a diagnosis of OCD, Obsessive-Compulsive disorder, which I've been living with most of my life. I hyperventilate throughout the entire day because I'm afraid that I'm going to lose the ability to breathe. When I speak for a certain length of time, and then I remember the need to breathe. So all of my breaths, inhale, exhale, are driven by that process of voluntary artificial breaths. I've been dealing with the breathing for close to 8 years. It's all I think about.
Okay, so there's just an example from Matthew's testimony about what it feels like to have treatment refractory OCD. Treatment refractory means that it's a form of OCD that hasn't responded to the standard treatments, including pharmacotherapy, psychotherapy, like exposure and response prevention. OCD is chronic. It's a kind of disorder that's characterized by intrusive, unwanted thoughts, urges, images. These cause a significant amount of distress. And the compulsion side that you've probably heard of, those are repetitive behaviors or can be mental acts. But the purpose of those are to reduce the anxiety that's brought on by the obsessions or to prevent something bad from happening. It's extremely time consuming and it's an entirely impairing way of being. Here's a schematic that summarizes decades really of research. We have really strong evidence at this point that is linking the symptoms of OCD to atypical brain function. We can synthesize all these findings in a meta-analysis like this and begin to sketch out parallel overlapping circuits. Really the early work, the substantial work focused on the yellow parts, there with the term NACC, which stands for nucleus accumbens. The nucleus accumbens is a part of the brain that is essential to the reward processing network. And that's ultimately how your brain reinforces a behavior, makes it more likely that it'll happen again, any behavior. So you can imagine what happens in a brain where the nucleus accumbens is hijacked. But then over the decades, the evidence broadened and cohered on what we now call the corticostriatal thalamic circuits. Those connect regions that are associated with that sensory motor experience that Matthew described, that connects to the cognitive, affective, and motivational processes in other parts of the brain. There's just so much complexity in how we integrate our behavior, even simple behaviors, with our thinking and our impetus to do things. And then, of course, the emotional or reward-based consequences of what we've done that'll reinforce that behavior. In OCD, it's like this integration is just out of tune. It's causing perseverative loops and stereotyped action and a great deal of distress. So this is why OCD qualifies as a circuitopathology. Okay, a circuit therapeutic approach is the general idea that we can intervene on any point in this system with electrical neuromodulation, and then we are fundamentally changing the underlying patterns of communication. It turns out that a core subset of what I showed you in the previous slide, the corticobasal ganglia circuits, they're particularly amenable to neuromodulation. So we can use electricity to interact with the communication to and from the subcortical nuclei, there I'm showing in brown, with the motor cortical regions and the prefrontal cortex, where we have our cognitive or thinking process, and the more ventral limbic emotional areas. I can get a little bit more specific with these subdivisions. They're topographically arranged in the brain along the dorsal ventral axis. That means top to bottom. So here I've coded them in blue for the motor aspect, like underpinning the rituals. Yellow for the cognitive control, the memory, the integration of those actions with past and future. And red, the motivational functions. There's that accumbens, the amygdala that you might have heard of, and the medial frontal cortex. And that's roughly segregating into our current targets for various indications for DBS. In blue for Parkinson's disease, the motor basal ganglia, the STN, the GPI. In yellow, DBS for OCD, which is targeting the connection from the ganglia into the frontal cortex. And depression, various targets that either directly or indirectly relate to those ventral limbic circuits. Very interdependent circuitry here. I think that's the take home. So if we intervene on one location, it actually resonates throughout the system. And that's why sometimes you'll notice that we use the same target for very, very different clinical presentations. It's also why a variety of symptoms within one disorder, like OCD, might be addressed with a single target. Deep brain stimulation, what's that? So DBS involves a surgical procedure where electrodes are permanently surgically implanted in the brain. This tiny little computer here, or what we call a pulse generator, is implanted under the clavicle. And then the wires go up under the skin of the neck and into the brain. Inside the brain, there is an electrode, and one is placed in each hemisphere. The electrodes generate current in the brain, can do that in all sorts of different ways. And for most therapies, we find the way that works, we turn the stimulator on, and we leave it on. To talk about that electrode or that lead in the brain, both of them bilateral, we usually use a cartoon like this one that I have up on the screen. So it's actually a string of electrodes. It's not just one electrode that are spaced about 1.5, 2 millimeters apart. So it gives us a lot of options about exactly where and how we're going to apply the stimulation therapeutically within a general target region, okay? So this is a little bit about DBS. It's FDA approved for Parkinson's disease, essential tremor, dystonia, OCD, not yet for Tourette syndrome or depression, but stay tuned, we're certainly working on that. So for all those indications that I just mentioned just now, there's really good evidence of treatment efficacy. Here are some results from a 2020 study from the Netherlands group. They're showing a whopping 40% reduction in the severity of OCD symptoms, here measured by a symptom checklist called the YBOC, which stands for the Yale-Brown Obsessive-Compulsive Scale. And notice that it's still holding after 1 whole year of treatment. Remember, this is a population of patients with treatment refractory OCD, so very significant severity and chronicity. Sick people get better and they stay better with this technique. So now, maybe not the not so great news, and that's that it's about a 52% response rate in this group. So that's remarkable, but we're still missing a lot of people. There is a significant variance in the treatment outcome, we'd say. And one source of variance is the targeting, getting that electrode in the right place for that particular person. So for the rest of the talk, that's what I'm going to focus on. I'm going to be talking about strategies to enhance the precision of the targeting within these known target regions, including this one, the ALIC, the anterior limb of the internal capsule, which is our target for OCD. So the internal capsule of the brain, there's that rainbow shape you see in the center of the screen, is like a superhighway of neural fibers that connects the basal ganglia, those nuclei that were in brown, on the right to the frontal lobe on the left of this schematic. The fibers, as they move from the middle to the front, they form a kind of ribbon shape, with the top of that ribbon connecting to upper parts of the frontal lobe and the bottom connecting to the more lower parts of that of the frontal lobe. So when a neurosurgeon places a lead in the ALIC, here's what it looks like on a stereotaxic schematic. You have a lot of territory there, a lot of heterogeneous territory to be searching for your mark. And actually, this schematic lets me tell you a story, a very interesting history about targeting the anterior limb of the internal capsule for OCD. And it involves that structure I commented on earlier, the nucleus accumbens, the reward center, which sits just below the ALIC, right there. So it made sense years ago when we were just beginning to explore this frontier of DBS for psychiatric disorders to target that area, because the idea was that must be the abnormal functioning of the reward system that is causing this maintenance of maladaptive behavior patterns. So the lead got placed there. But then over the years, the clinicians were making trial and error adjustments to where they were stimulating along the lead and just watching patients to see if they'd get better. If they weren't getting better, they might make an adjustment and so on. And lo and behold, over the course of several years, they discovered that people were actually getting better on the upper contacts, not on the accumbens contact. And that is what ushered in an era of exploring this very complicated ribbon of fibers as the potential target for OCD. I tell the story just to underscore the nature of the discovery narrative. where we start with a functional idea and then we work with patients to understand what's working, what goes on, and make adjustments as we go. And then we learn. We learn from what happens and go back and update our model. I'm going to show you what that looks like. I'm going to go back to Matthew's story now and give you 2 examples of times when he was stimulated in different places around that target region. First, we're going to watch when he was receiving stimulation at that lower place, a little bit closer to the accumbens, here in the red circle.
I mean, it's okay. I feel kind of like in my heart, like I don't know. Oh, yeah. Like a little, yeah. I'm like laughing now. Does the laughing like go away or? Yeah. The first activation of the system is usually a little hypomania causing, especially this contact. But we're going to move to another contact soon. Like I feel like I want to dance. I feel like I want to run on the street. Like, oh yeah, yeah. I feel very energetic more than normal. But naturally, or like you're on drugs? Like a little like I'm on drugs, but like it's not bad. If you feel great, you might be better able to control your OCD as well. Yeah. But it's not what we're strictly aiming for.
Okay, and let me underscore a little bit what Dr. Figuet just pointed out, that maybe that kind of mood boost that he was receiving through stimulation and the most ventral contact could be useful in certain situations, but it's not really what he is aiming for as a clinician in terms of the perceptive symptoms of OCD. Let's try a different contact. A little bit higher, the teal level.
It's going to be left 3C, right 3B. I'm not even compulsively related. It's almost gone. Yeah, definitely less. So like, but that was a good setting. The compulsion is virtually gone. Like I'm not compulsively breathing and right. Yeah. Not too up, but definitely way more down. Better. Yeah. Wow. It seems that like the contact is the best one, which is the third one, because it doesn't give you this up, this sort of mania. Yeah. This is like a whole new look on life. This is crazy. Right, so here's the phone.
All right, so thanks to Matthew for sharing that experience with us. It gives you a sense of just how exciting and fascinating it can be to work on this particular frontier. But I want to say that many people don't see an acute behavioral effect like this, or sometimes it's ambiguous, or it can even be absent. So relying on this kind of subjective experience to optimize the targeting is just not sufficient. We have to find another approach, and we're starting to hone in now on this target. I'll put the little green dot here on where we're aiming, just a little bit above the accumbens area in the ribbon of the ALIC. And then down here, you can see that the little green and yellow dots are showing targets more in the accumbens region that were actually successful or not successful for the treatment of depression. So having established this target in the ALIC zone, we're good to go, right? We have our sort of feature to chase, but it turns out there's still work to be done, because if you look at that target location in a different way, you can see how the same stereotaxic coordinate can be very different in terms of other features. Let me show you an example. So here I'm showing 12 different patients, and the colors for each of these patients are showing different distributions of white matter fibers of passage that are transversing that stereotaxic target. So even though the target according to the cell types and the landmarks, is the same for each of the patients, the fibers of passage that are moving through that area are very different for different patients. What's a white matter fiber of passage? I'm referring here to these long haul neural cells. They're insulated cells that can pass information very quickly from one part of the brain to the other part of the brain. They're passing through the target zone on their way to and from different parts of the circuit. So appreciating these white matter fibers as an aspect of targeting in addition to the stereotaxic coordinate was a real game changer for DBS across indications. And this is true for ALIC, DBS for OCD as well. The field, in fact, is now converging on a set of fibers that seem to be related to optimal outcomes for OCD. It's still early, but a meta-analysis, which is a kind of study that combines findings from multiple groups, points to the importance of this sort of pink salmon-colored bundle that moves toward the frontal lobe. And then my colleague, Dr. Smith, pictured here, recently published an analysis of our patients' therapeutic targets, and he found that their response-defined optimal location for stimulation was actually consistent with that putative connectome blueprint that I'm showing there. Moreover, he reported that his fiber score, which is the amount of overlap that his cohort had with the meta-analytic finding, correlated with improvement at our site. So what we're learning is that tractography is important, and that might explain why our response rates are increasing now from 50% to between 50 and 75% response, but still with a lot of variance in there. So this work is a major priority at our center and a primary goal of a clinical trial that I will discuss a little bit later in the talk. This tractography guided targeting for deep brain stimulation was an essential insight. It was pioneered by Dr. Helen Mayberg, who's also the director of our center. She actually studies a different target for DBS called the subcallosal cingulate, or the SCC, to treat depression. And in many ways, what we're doing in OCD is a recapitulation of her discovery narrative for the depression target. Started in the 90s, really, when she, just as we did, established a functional model of the brain pointing to hyperactivity in a region pictured right here called the subcallosal cingulate, or Brodmann area 25, in people with depression or when profound sadness was induced. And this was reversed with various forms of successful treatment. So she then had a game-changing idea that she could use deep brain stimulation to cause a temporary disruption to that region, and it worked. It worked in profoundly ill people, some, not all, but it worked. And that really ushered in an era of deep brain stimulation for psychiatric disorders. It turned out in her case as well, as they went to refine the targeting, that the stereotaxic coordinates weren't sufficient to explain why some people got better with treatment and others didn't. And so the hunt for the sweet spot began, and that led Dr. Mayberg to another major innovation. She started to work with my colleagues, Dr. Patricia Riva-Pose, Dr. Kisung Choi. Actually, Dr. Choi was also an RSAD recipient at one point or another. Together, they were looking at these white matter fibers of passage that I just described earlier, and they noticed something really important. They noticed that all the people who got better with SCC-DBS had something in common. That's the blue color that we're seeing on the screen. What they had in common is that the therapeutic contact for DBS was placed right at the confluence point of these three superhighways, these fiber bundles that are traversing the target area. And if you weren't convinced already, they ran another experiment where they imaged people who weren't getting better with the treatment, and then they changed the target so that it was at that confluence point, and lo and behold, those individuals started to get better. So this was really strong confirmation that what we need is a connectome blueprint to define the optimal target engagement in an area. Moreover, as the team got better and better at this tractography-guided surgical targeting, patients got better faster, they stayed better longer, and the responses were more robust. So refinement in targeting really matters for the longevity of the treatment. That said, around the same time, a multi-site RCT was halted for futility, which means there wasn't a fast enough treatment effect at the start of the trial to discriminate between the treatment group and the control group. And one really clear reason for this slow start in the big trial was that we had insufficient target engagement at various sites. So this story is clearly not over. In fact, that's when I began to work with Helen's team. I came in with a mandate to close that gap between the tractography models and the real-time effects so that we could disseminate an objective protocol for targeting to various sites and run an effective clinical trial, because then you can use some kind of method to confirm that you have achieved optimal targeting for an individual. Moreover, we need some techniques that can account for how the brain might change over the course of treatment, including brain shift after surgical implantation. And like I said, unlike movement disorders, that acute symptom response is just not sufficient in psychiatric DBS. We can't rely on that to be the way that we select the optimal parameters for the device once it's been implanted. Wouldn't it be great if we could just go directly to the brain over the course of care to assess whether we have target engagement and to compensate as needed with parameter adjustment? That is exactly what we're proposing to do using electrocortical measures and a technique we call perturbation mapping. I'm going to take one slide now to just build some shared language amongst us about electrophysiological measures. So I've been talking a lot about these white matter connections and the nuclei inside the brain, but the cortex is really where we humans excel. You might be surprised to learn that the cortex, which you hear a lot about, is actually a very narrow sheet or a shell. It's only about 2.5 millimeters wide, and it has lots of layers in it. And one of those layers contains a really big neuron called a pyramidal neuron. The pyramidal neurons, pictured right here, they're all lined up side by side. So when they fire together, we get a little current running in the direction of that action potential. Now, tiny currents create what's called an electrical field potential. Electric field is sort of like the amount of tug you'd feel on a charged particle to be moved by the current. It's remarkable that we can actually measure those fields that are caused by currents in the cortex from the neurons firing on the surface of the head. Just as a convention, we typically show these fields in terms of positivity and negativity, polarity in red and blue, respectively. And in the field, we often represent that with a circle, as if the circle is the head and we're looking down on the head from above with the nose pointed up. So what's incredibly cool about this is that we can capture the voltage pattern on the head surface, and then we can also make some really good guesses about where the currents are coming from in the brain that's causing those voltage patterns. In other words, we can measure from the outside what's going on the inside of the head. And we're exploiting these properties to build some maps of how the brain responds to a volley of stimulation in the target region. Here's how it works. The patient comes to the lab, they get all wired up, and then we fire the DBS with tiny pulses. Between the pulses, we wait just long enough to see how the cortex responds, and we repeat that 500, 300 times, and then we average across all of those stimulating events. By averaging across them, you discard anything that was happening in that moment that didn't have to do with the stimulation, and you retain only the cortical activity or the electric fields that are caused by that volley, by that pulse of stimulation. The result is so remarkable. It's coherent, it's mysterious. It's a pattern of voltage fluctuation on the scalp surface that reflects, as I said, synchrony in the cortical activity that results from stimulation. And it lasts 100 to 300 milliseconds, so about a quarter of a second. The cortex is responding to this pulse of stimulation. It's a technique that's been around for a long while, but the application is just now starting to be appreciated, including by the BBRF, who funded research in this area. Our goal at that time was just to examine the design properties of the signal to see if we could apply this strategy to the target engagement problem. Maybe you're already developing an intuition for how we might do that. So by design properties, I mean, what is it about this technique, this signal, this seemingly abstract, that has what we need to engineer what would be a patient-level brain readout that's somehow informative to DBS care? And here's an example. It turns out that the stimulation or the voltage map is very sensitive to where in the brain the stimulation is applied. So here on the screen, I'm showing perturbation maps from 3 different DBS targets: SCC for depression, ALIC for OCD, STN for Parkinson's disease. And you see that each one produces a signature pattern, indicating that depending on where you start in the circuit, where you perturb the circuit, you're going to initiate a different cascade of events in the cortical circuits and connections of the brain. Moreover, we don't only have variance on the level of location in the brain, but we also see this pattern really captures something special and unique about individuals. So I've been showing those voltage patterns projected on a 3D image of a head. Now I'm going to move back to that convention I was saying we use in the field, where we imagine that your head is a sphere and we're looking down on the top of that sphere of your head that's been collapsed into 2D with your nose pointing to the top of the screen. And what we're looking at here are 6 patients, each of them receiving stimulation to the same target, the SCC. So you might see some of the same canonical pieces, features that we have come to associate with SCC perturbation. But just like faces, where we all share eyes, nose, mouth, the perturbation maps are still quite consistent but individualized. So we can recognize even whose perturbation map we're looking at return visits. Just as a reminder, down in that inset, I've put a different location of the brain, the STN, so you can really appreciate the sameness and the differences. All these movies and descriptions are really just to say that this is a robust signal measure of cortical response to stimulation that is sensitive to the key parameters that are relevant to this treatment. And our work now is to decode what these location and individual differences mean so that we can individualize the DBS parameter optimization according to that readout. We just need to learn how to read these maps to confirm optimal target engagement for individuals. So for the last quarter of the talk, I think I'm going to take you on a spin through some of the first steps that we've made to accomplish that goal. Our first focus was on depression, where we learned to characterize the stereotypical patterns that would arise from stimulation of that target region. We learned that it was very reliable over time. And we learned that the waveform was likely an artifact of something called phase alignment, which means that the waves of a certain frequency band just suddenly crashed at the same time. That's phase alignment in the frontal theta band, which happens to be a frequency band that's long been implicated in depression and depression treatment. Something really important is that these features are sensitive to millimeter scale changes in the location of the pulse within the target region. So we're able to feel around in there and make some determination about where we're at based on how the cortex responds. A really high priority for us is evaluating whether or not these cortical responses can be used as a proxy measure for the stimulation of those white matter fibers that I described earlier in the talk. That's so important, just given the centrality of tractography in the discussion of targeting right now. It would be a real game changer if we had a real-time readout of how well we're hitting those fibers of passion or fibers of passage. I sort of think of an electrician who's pinging a circuit. But you know, we've tried for several years to really make that connection. So far, the direct correlations between the two have been pretty modest, but we're now exploring some more dynamical approaches to extracting the temporal features that might better explain that association between white matter and the voltage patterns. Similarly, another really fruitful approach had us ask instead how much these perturbation maps might reflect differences in white matter affected by the current that is being put into the system. This is a collaboration with the McIntyre team at Duke. My colleagues, Brian, had an idea that he could build some models that would specify the way we might bend the current, essentially by adjusting the DBS parameters to theoretically perturb just one fiber bundle at a time. So the top two would be examples of the parameters that would hit just the cingulum bundle, and the bottom two were attempting to hit just the forceps minor in red. And indeed, when we did that, we got evoked responses of different shapes and voltage, spatiotemporal voltage patterns of very different shapes. So we're still trying to figure out what those differences represent, but have clearly established a relationship between the head surface, voltage, change rhythm, and exactly what cells we're stimulating within the target region. So we have this experience with depression, and we are poised now to accelerate the discovery process for OCD. This is our current trial. It's under the direction of Dr. Figuet and Dr. Choi, and it is essentially recapitulating what Dr. Mayberg did over a decade or a decade and a half. We're now halfway through enrollment of people with treatment refractory OCD, and we're still open for enrollment. So if you think this treatment could be right for you or a loved one, don't hesitate to reach out to us for a screening. We're aiming to track 20 patients over time in treatment with ALIC DBS. And the aim here is to validate a connectome blueprint, but also my colleagues are doing some work on selective stimulation and behavioral models, and my team is collecting the ALIC perturbation maps at different locations and settings and time. In fact, this was the work that was seeded by the BBRF NARSAD Award, which was a pretty big risk that I appreciate that the organization took, because at that time, we didn't even know if there would be a signal if we attempted to build a voltage map or a perturbation map from volleys of current into the ALIC. Oh, but there is, and I would really like to share those preliminary results with you from our first 10 patients. So these movies that I'm about to show here are how the cortex responds to pulses of stimulation to the anterior limb of the internal capsule. The right shows what happens on the head surface when the pulse is delivered to the right hemisphere and the left to the left hemisphere. We have never observed this pattern before, so we're going to need a little bit of time to decode it, but I can already point out some really striking, interesting divergence from the SCC map, which is pretty symmetrical. This map sort of oscillates back and forth, left and right, and I wonder what that might add to our notion of the optimal target. Here's some work from my very talented collaborator and signal analyst, Davide Momi. He's working to transform those patterns on the head surface into the cortical architecture, as I described before. And here in this movie, we can see those lateral effects again, but I would also ask you to sort of peer in between the hemispheres if you can. And what you see there is the orbital frontal, the ventral, the lower part of the brain, and the frontal lobe and the ventral medial cortices lighting up. Very important question here is how these hotspots that we're seeing might reflect the cortical termini of the fiber bundles that are currently in our blueprint. Alternatively, these patterns might better describe functional network engagement. So here, Dr. Momi is using canonical parcellation of the cortex. They're described as the YO networks, 7 different networks. And then we ask which functional networks are engaged when we pulse the ALIC or nudge the ALIC with a little bit of stimulation. And then, of course, the question is, does this reflect the circuits that I described earlier as being involved in OCD that I started us off with? And our preliminary analysis says yes. The anterior features of the default mode network representing I would say the greatest activations in teal on the schematic below, but the second largest network activation is in the salience or the ventral attention network, which is roughly related to the red pieces in the OCD map below. And of course, it's also very possible that the cortical evoked response to ALIC stim is not a very good proxy for functional networks or structural networks. Maybe it could be its own new dynamical property of the brain that we're able to capture and observe in near real time, and could be harnessed and important for optimizing targeting, understanding the pathology of the circuits. Really, the most important element of this technique is, I think, that it's not a leap to imagine seeing this kind of strategy in the clinic. It's a non-invasive measure. You can get it any time over treatment, and so we can imagine our patients coming into the clinic to get these measures while they're getting adjusted, and we're essentially listening in on how their brain responds while we hunt around for the optimal parameters that will generate those electrocortical features that indicate optimal resonance in the circuit. This would eliminate a lot of guesswork and maybe up that percentage of people who are willing to undergo the procedure and get back to living their lives. It's such a pleasure to give this lecture, share some of the rationale for why it's reasonable to think of OCD as a circuitopathology, and how optimal targeting of brain implants becomes really the critical and also very complex aspect of the need for innovation in this area. I used the case study of DBS for depression to show a roadmap for testing innovations, including behavioral strategies to target, tractographic strategies to target, and then I ended with our leading edge, which is really your first look into an emerging technology using electrocortical mapping or perturbation mapping to characterize and define how an individual's brain responds to stimulation in a target area. So now I want to say thank you to BBRF and my host, and thank you to the center and my lab. And I would love if we have some time to have a brief discussion.
Allison, we do have a little bit of time, and thank you for an outstanding presentation on a complicated, very complicated area of research and clinical work. So thank you for that. The one question is, you described the work that you're doing on OCD and the work that Helen Mayberg did on depression. What other conditions do you see this type of approach as potentially having an impact?
That's a great question. Well, right now, the diagnostic indications for DBS include Tourette syndrome and various motor disorders, movement disorders, dystonia, PD, essential tremor. And what's wonderful about having all of these different treatment for all these different indications under one roof is that we can borrow insights from one indication and apply them to another. So while movement disorder, DBS for movement disorder has been around for some time, and in some ways they're leaps and bounds ahead of where we're at with psychiatric DBS. And yet we've learned so much, for example, about tractographic targeting in psychiatric DBS, in part by necessity, right? Because in psychiatric DBS, we don't have the motor symptom to target and tune the parameter optimization. The psychiatric symptoms change on the scale of weeks to months. So that necessitated a certain kind of innovation in the psychiatric space that now can feed back into the motor area.
Right. Great, thank you. I want you to just tell us a little bit about how refractory OCD would be defined. What types of treatment should a person have had that didn't work to then meet that criteria of refractory?
So you would have to have failed, I think, 3 psychopharm treatments, as well as empirically supported psychotherapy. That would be exposure and response prevention therapy. This time, there's also a chronicity factor. So treatment refractory would also imply that this is a severe and long-lasting form of OCD.
Okay.
Yeah.
And obviously this is an invasive treatment, which for some people really, is life-changing in such a positive way. How long after the surgery, the procedure, how long, what's the rehab period? How long does it take for the person to be up and about and sort of back doing their activities?
Oh, it's such an interesting question, and another frontier of development in this area, really, Jeff. So response times, response latency is variable. And of course, we want to know why. But that aside, this notion that the circuit perturbation or the DBS treatment sort of opens a window of plasticity in the sense that patients can actually return to forms of therapy that hadn't worked for them in the past, or had worked, but they weren't able to maintain their treatment gains, and try again. And they report that with the device on board, they're better able to engage, for example, with exposure and response prevention therapy. And this is why it is essential that when we bring these directive brain interventions, these devices on board, we're ready also to provide rehabilitation services that are informed by this unique situation that the person finds themselves in with an implanted device. So that is exactly, you are exactly pointing to a big area for innovation where we can study this and understand better how to more closely integrate the direct-to-brain treatment with DBS with targeted rehabilitation practices to enhance really the lasting benefit of both.
Great. Thank you. Well, I want to again thank you for your ongoing research, for the presentation today. In many ways, it sounds like science fiction, but it's real science. Mm-hmm. So thank you, and I look forward to having you back again in the not too distant future to give us an update on all of this important work that you're doing. So thank you so much.
Thanks, Jeff. I look forward to that.
Yes. I also want to thank everybody who joined us today and ask people to consider making a donation to BBRF. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientist. To make a gift, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it, please visit the events and webinar page on our website. I'd also like to invite you to join us again on June 11th at 2 p.m. Eastern Time, when Dr. Catherine Burdick, the Jonathan F. Boris, MD, Distinguished Chair in Psychiatry and the Vice Chair for Research in Psychiatry at Brigham and Women's Hospital in Boston, Massachusetts, will present Neurocognitively Defined Subtypes in Bipolar Disorder, a Path to More Personalized Treatments. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Take care.